academic unit for psychiatry of old age. It's an absolute pleasure to be here today at one of our NARI Tuesday seminars, which I think are always really fantastic. We get such diverse presentations here, organised beautifully by the NARI team. Before we get into anything, I want to start by please acknowledging the traditional owners of the lands on which each of us will be meeting today in this sort of virtual space. I'd also like to pay respects to the traditional owners across lands and waters across Australia, and respects to elders past, present and emerging, and any Aboriginal or Torres Strait Islander people who might be attending us with us today. I'll start with a little bit of housekeeping regarding Zoom. I mean, we've all attended a lot of these now, but I think it's still important to give us a bit of context on how to go about things for this particular meeting. And we want to hear from you. So if you've got some questions, please feel free. We'll open the Q&A a little bit later in the chat function. Feel free to put questions in there. Feel free to put a hand up when we get to question time at the end. Love to hear from you, and we'll get to as many questions as we can in our hour today. Please just keep in mind that we're recording today, so if you do put your video on and ask a question, you will be shown in that video there. Okay, so today we'll be hearing from Associate Professor Albie Elias, and he's a Clinical Associate Professor with the Academic Unit for Psychiatry of Old Age within the Department of Psychiatry at Melbourne University. He's also the Director of the ECT Services and a Consultant Psychiatrist at Western Health. His PhD investigated the risk of Alzheimer's disease in Vietnam veterans with PTSD using both amyloid and tau PET imaging markers, and he received a project grant relatively recently from Dementia Australia for the study that he'll be discussing today, which is something that I think many of us will have a bit of an interest in, and that's intermittent fasting for dementia risk reduction. And he's going to give us a current status update. So without further ado, I'll hand over to Albie for his presentation. Thank you, Albie.
Yeah, thank you, Catherine, and good afternoon, everyone. So to start with, as Catherine mentioned, I acknowledge the funding support from Dementia Australia for a pilot study on intermittent fasting in dementia. I actually presented this topic in the annual symposium of the Academic Unit of Psychiatry of Old Age in December last year. And when I was asked to present this topic in this seminar, I was initially thinking whether it's too short gap to present this again. Would it be boring? But you wouldn't believe quite a few things happened in the last 1 or 2 months. And, things always keep happening. You might have seen this study that appeared in media, heavily infiltrated social media, and many people contacted me. The study that suggested increased mortality, cardiovascular mortality in people practicing intermittent fasting. Obviously, this caused a lot of confusion and commotion among people practicing fasting. But I will talk about this study towards the end of my presentation. Some methodological issues with this study. Secondly, this review paper just appeared a few weeks ago, and this paper discusses so many insightful findings related to middle age, and some of them can even challenge our conventional understanding about aging. So the bottom line is middle age is a very critical phase in the trajectory of aging. Unfortunately, the changes happening in middle age are vastly understudied, and these sort of changes go under the radar. So we will see how these changes shape future brain and the diseases that we see in old age. We often discuss about the neurodegenerative disease in old age, but it's also important to see how middle age changes shape this trajectory of aging. So there are certain evolving concepts about aging, and one is, as I said, the middle age represents a big shift in brain aging. So the middle age is taken conventionally between 40 to 60 or up to 65 years of age. And it's actually this paper, the paper I mentioned, personally, I felt a bit depressing because it generates a strong doubt, a question whether, the preventive interventions that we believe might be beneficial in preventing this neurodegenerative disease of old age, the middle age itself is late for these interventions. I'm not too old, but I'm not young either. But I should say, uh, better late than never. So the question is preventive intervention, when we should start. With the emerging findings, there is a strong, compelling point that we should start this intervention in middle age rather than waiting for older age. One phenomenon with aging is it's a non-linear, some aspects of aging show non-linear relationship. I will explain what it is in the next slide. And then I will talk about how intermittent fasting can modify some of these changes happening in middle age and how that might be beneficial in reducing the risk of dementia as a hypothesis. Then I will talk about the current study of intermittent fasting and dementia, which is on the flagship of the Academic Unit of Psychiatry of Old Age at the University of Melbourne. So the linear relationship is when you plot one variable along y-axis and the other one x-axis, those two variables correlate with each other, and it shows the same direction. And by linear relationship, we mean that an outcome at one point can be predicted from preceding events. So it's a perfect correlation, and it's the same direction. So we know in which direction it is going. Whereas if you look at the nonlinear correlation, the things change. They are not in the same direction. And for example, the events here can't be predicted from the preceding events. And when it comes to aging, we know that tissues grow, organs grow up to a certain age, and the cognitive functions mature, and then they get plateaued, stabilized, and then they show decline. So now we know how the trajectory of aging, but as a descriptive function, if we look at the changes in young age, then at the late age, they are not proportionate. So we will get no clue of what would happen in late age by looking at the changes in young age. So it shows a non-linear relationship. So many recent findings suggest this non-linear aging in many ways. For example, what causes to this non-linear aging? So one is different organs age differently. For example, like brain age, we have heart age. Now, cardiologists can estimate one's heart age based on the parameters that we enter into an app. For example, our body, BMI, our cholesterol level, blood pressure. So it's highly individualized. And this is based on machine learning and data mining. So this represents a kind of precision medicine where we can predict our heart age from all these variables. So heart age is different, probably different from brain age, and different organs age at different rates. Now, even one organ shows aging at different rates across lifespan. And different parts of the same organ age at different rates. So there is a lot of heterogeneity in the aging process. across organs, across lifespan, and across individuals. So in human brain, the gene expression and the protein synthesis related to the synaptic function of the brain plasticity, these function undergo accelerated changes in the middle age. So the rate at which these things happen is not uniform across lifespan. And it is now proposed that middle age is the phase, the time when the changes that account for individual differences in late age occurring. So that's the importance of middle age, because I'll explain some of those changes happening middle age. Now, coming to the Alzheimer's disease pathology, this also shows a kind of non-linear relationship. For example, the beta amyloid protein, which is forming the cardinal lesion of Alzheimer's disease, the plaque. So the beta amyloid deposition, it begins approximately 15 to 20 years before the symptoms of dementia start. So that mainly happens in the middle age. So that building up happens, it can start anywhere from 40 years, depending on a few other factors. For example, the presence of apolipoprotein E4 allele. But then it builds up in the subsequent in the following years to culminate in Alzheimer's dementia. And then the brain structural changes follow that. It also has got a different trajectory, and then the cognitive decline starts later, and that also may not perfectly correlate with amyloid burden in the brain. So these things may dissociate. People can have significant amyloid in the brain, but without dementia. So what I'm saying is that the amyloid deposition starts in the middle age, though we see dementia much later. So this is also a non-linear process during aging. Now, as I mentioned about amyloid deposit and neuroimaging and findings and cognitive function, which may not correlate with each other perfectly, the physiological process need not reflect in the neuroimaging findings. For example, in the part of the hippocampus, the dentate gyrus, the atrophy in that region need not linearly correlate with the physiological functions in the hippocampus. For example, the field activity and also the cognitive function associated with that activity, with that region. So there are separations or dissociations between among these variables. And memory, the reaction time and the processing speed, they also show an accelerated decline towards the end of middle age. And some other functions, even show decline even in the before that, even after 40 years of age. And this is joins some of the psychosocial variables. Middle age is a time marked by many psychosocial events. Example, loss of parents, change in career, or retirement, and many other things. And in women, the menopause and postmenopausal changes all add up to middle age changes. And also we find that the benefits of repeated practice test, they disappear after 60 years. So middle age represents so many changes, and these changes then progress into old age. But the proposal is that the disease we see in old age actually have strong roots in the middle age and the changes happening to the brain during this phase. Additionally, some of these biochemical pathways, I will explain what mTOR is, and the brain-derived neurotrophic growth factor synthesis, all are disproportionately affected in middle age. And this gene allele, apoprotein E4 allele, which carries strong risk for Alzheimer's disease, its presence causes cognitive decline, particularly in episodic memory around 60 years of age, and that also happens in middle age. Then connectomes, the neuronal networks, for example, there is default mode network. You might have heard about attentional network. So these networks function properly and adequately when they are separated from each other, but when there are within the network dense connections. So the anatomical distance between these networks peaks around 40 years, and that allows this network to serve special functions. So that specialization will be lost, or it will decline in the next 10 or 20 years, and that also happens in middle age. Then several epigenetic changes happen in middle age. So the epigenome is not a part of the genome that codes protein, but epigenome can influence gene expression. So these epigenetic changes, by modulating several cellular reactions, are related to multifactorial disease that we see in old age. And few examples of epigenetic changes are DNA methylation and post-transcriptional histone modification. So these epigenetic changes influence protein synthesis, and they also influence disease expression. Now, coming to how intermittent fasting can affect these changes that happen in the middle age. So one phenomenon is called autophagy. The name itself suggests, the meaning is self-eating. So essentially it means that the content within the cell can be degraded or even destroyed. The cells destroy its own content. But going deeper, we can see that it's a form of cell recycling. It's a kind of housekeeping activity. So what happens during intermittent fasting is depletion of glucose, and the cells have machinery that can sense glucose depletion. Once it happens, then a membrane is formed and it gets elongated and it encircles non-essential components of the cells, which include some proteins. So all are encircled into a sac, and then it fuses with the lysosome, which lyses this membrane, and then the proteins are degraded into individual amino acids, which are the constituents of proteins. In this process, the proteins are degraded, and then they are removed. So this can also involve pathogenic proteins, so the proteins that are related to neurodegenerative diseases. So autophagy is a process where cells can recycle its own products, and it's a normal physiological mechanism. It's present in every cell, and it's a highly preserved mechanism during the evolutionary process. And impaired autophagy is associated with aging, some cancers and neurodegenerative diseases, because cells cannot repair themselves. And interestingly, we find that intermittent fasting can promote autophagy because the glucose depletion is one trigger for this phenomenon. Now, mTOR pathway. So mTOR stands for mammalian target of rapamycin. It was originally detected from a fungus, and it's a protein kinase. It's like a molecule that receives multiple upstream biochemical pathways. It's like a runway in an airport where flights from many directions land on. So mTOR pathway receives transduction signals from multiple pathways, and then it regulates protein synthesis, and it is again preserved in the evolutionary process, one of the most ancient molecule found in almost all eukaryotic cells in mice and humans. And mTOR is related to protein synthesis, but its activity can be regulated by glucose level and also hence insulin levels, oxygen, cholesterol, and amino acid, and many more. But what happens is, once this mTOR pathway is activated, it can inhibit autophagy. And mTOR activity is associated with effects of aging. And excess mTOR activity is also related to some cancers. Now, we know that metformin can inhibit mTOR pathway. I should say metformin is a wonder drug, and reducing metformin is just as an anti-diabetic medication is misleading because it has other cellular effects. And one example is inhibition of mTOR pathway. And in some studies, metformin showed some anti-aging effect, anti-cancer effects. And then intermittent fasting also shares the same effect when it comes to its effect on mTOR pathway. So this is a representation of mTOR activity. It is triggered by various changes in glucose, insulin level, sometimes infection. I mean, incidentally, during the time of COVID, some researchers investigated how the virus replicates in cells, and there is a strong postulate that the viral particles use mTOR because mTOR is associated with synthesis of proteins, and the viral proteins also can replicate. And I have seen some studies on intermittent fasting which showed benefits during COVID infection in terms that it reduced severity of the infection, ICU admission. Anyway, that's a bit incidental. So by influencing mTOR activity, intermittent fasting can reduce autophagy. It can reduce the risk for several neurodegenerative diseases. So it has got, the intermittent fasting has got a very strong theoretical framework and a strong mechanistic aspect. But our question is how much empirical evidence we have got to support intermittent fasting in human diseases. Before going into that, briefly, intermittent fasting, what it is, essentially, it is a longer interval between meals without entering malnutrition or other harmful effects. So, according to different studies, the maximum period of fasting is traditionally taken as 24 hours. Beyond that, we might see biochemical changes that are seen in starvation, which could be harmful. So we don't go beyond 24 hours, but although there are studies that tested the effects of prolonged calorie deprivation, for say example, up to 72 hours, but that's not usual or common. Some people practice fasting for 16 hours or 18 hours, then the remaining window for eating. But make sure that we don't end up with malnutrition. So it is not a type of the diet, it's an eating pattern. And in many studies, intermittent fasting was compared with ad libitum diet, which means eat as we wish, or there's no limit, or regular diet. Ad libitum means as we wish or as it is available. So we know that intermittent fasting is not something new. I mean, humanity practiced fasting for millennia. I mean, it has been part of humanity since our evolution, and it is highly preserved. practice, not only in humans. It is seen in nematodes. We see that for months in polar bear because they have a very thick fat pad. And during nutritional deprivation or glucose deprivation, they use fat pad as a source of energy. And intermittent fasting has significant anti-inflammatory effects, antioxidant effects. All these have been tested in studies, but mostly in animal research, because during fasting, I'll show in the next slide how it works. There is reduced oxidative process, and fasting induces adaptive stress response in cells. So it is like this with fasting. There is a stress, but it's an intermittent stress with intermittent fasting. So with the stress, cells go into an energy conservation mode because the cells can detect reduced amount of glucose and other nutrients, and cells try to preserve energy, and the production of proteins is decreased as a part of energy conservation. So that leads to adaptive responses, including autophagy, when these damaged molecules are removed from cells, and that also leads to cellular repair. And when we get food next time, it restores cellular homeostasis. So with this stress response system, cells attain better fitness. It's something like as we are tested, we become stronger. So cells undergo this cycle repeatedly, and that stimulates its adaptive response. And this increases cellular fitness, especially when cells face degenerative diseases. Now, coming to the basic pathology of Alzheimer's disease, I'm not going to the details, but the most important lesions are beta amyloid plaques and tau protein tangles. And then vascular lesions also contribute to Alzheimer's dementia and, of course, to vascular dementia. There is a component of inflammation and oxidative damage. Now, as you can imagine, during fasting, the cells reduce the productivity. In mitochondria, the production decreases. So in mitochondria, which is the powerhouse of cells, when the glucose combines with oxygen, free radicals, the oxygen-free radicals, will be generated, and they can bombard the nucleus, causing DNA damage. I mean, normally our body has oxidative defense mechanisms, which are mostly produced by metallic enzymes. But when the free radicals amount exceed the normal defense system, that can cause DNA changes and mutations. But with fasting, that risk can be reduced. Now, coming to the empirical evidence, I should say these are mostly derived from animal research, and in animal models, fasting was associated with improved cognitive performance and improved motor coordination, learning and consolidation of memory compared with animals fed with ad libitum diet. Along with the improvement in cognitive function, there was also there were changes in hippocampus. The hippocampal layers become thickened, and intermittent fasting was associated with increased production of brain-derived neurotropic growth factor. But the limitation is, this is very important to keep in mind, animal models are quite limited. I mean, we cannot replicate Alzheimer's disease exactly. The big problem is the time scale, because we know in humans, the pathology of Alzheimer's disease, it mounts and it builds up over decades, and we cannot create animal models for such a long time scale. So it is limited, but it can set some theoretical foundation for future human trials. But having said that, there have been studies in humans on intermittent fasting. It has been studied in people with obesity, high blood pressure, rheumatoid arthritis, increased cholesterol levels, and preliminary evidence suggests that intermittent fasting was efficacious in reducing cholesterol, obesity, and inflammatory reactions. Then coming to safety and how much people can tolerate, it's largely safe, including in older people, without any serious adverse events. The common adverse reactions included fatigue, and sometimes people can get irritated. They may show mood changes. But as time goes on, people adapted or adjusted with this new lifestyle. And they then practiced intermittent fasting for months. Some trials tested this intervention for 6 months, and some are looking at even at 2 years. Intermittent fasting is not suitable for everyone. I mean, for people who have type 1 diabetes mellitus on insulin, they can go into severe hypoglycemia. We cannot recommend, or we need thorough medical evaluation, or can be done with strong medical supervision. It's not good for people with several other conditions like eating disorders or low BMI, emaciated people, those with cardiac arrhythmias. We need to be careful in recommending intermittent fasting in these conditions. So it's not for everyone. And another effect of intermittent fasting is reduction in lean body mass and in muscle mass. But this can be mitigated by resistant training. And then we found that the lean body mass reduction can be corrected. Now, coming to the study, this study, first of all, this was not peer-reviewed. It was presented in a conference. It's a conference abstract. And what the investigators did was they called this participant, just 2 phone calls, 2 days apart, and they asked about the diet and the eating habits. And they divided participants in 2 groups. And the one group practiced intermittent fasting based on this very short and 2 phone calls, and the other group that did not follow intermittent fasting. And they found that cardiovascular mortality was higher in the group that practiced intermittent fasting. Now, I reviewed the whole abstract, and what is concerning is one, selection bias. I mean, there is high possibility that people with certain conditions, for example, a risk for stroke or heart attack, might practice intermittent fasting knowing that this might help. So there is in statistics, this is called reverse causality, because it's actually these cardiac conditions that lead to intermittent fasting, not the other way. So when they start fasting, to begin with, they have these conditions. So the selection bias was not addressed in the study, or not even looked at. And another problem is when we say people practice intermittent fasting, there is a high, it's like craving for food. People fast for hours, say 16 hours. And after that, there is a notion that, I practice intermittent fasting, I cleaned it up, then I can eat. And there is inherent and instinctive behavior that, we always seek food, and appetite is strong, there's food craving, and one might end up with compensatory eating and it can involve anything, rich in calorie or fat and things like that. So sometimes it can become intermittent eating rather than intermittent fasting. So in this study, there's no clue or there's no information about what people ate in between periods of fasting. So there are not much information about other results. So this study, unfortunately, circulated in WhatsApp and social media. Many people contacted me and asked what I'm going to do. But I should say, I don't think that the study will stand against the wealth of information that previous studies have generated about intermittent fasting, its benefits, and its widespread biomechanical aspects. So these are the methodological issues. And now coming to our study, the Academic Unit of Psychiatry for Old Age. This is going to be a randomized controlled trial of intermittent fasting in people at high risk of dementia. Now, ideally, to measure the effect of this lifestyle intervention, we need a long time scale. We need to study this intervention for years and years in a large sample of participants in order to detect a meaningful difference or a meaningful result. So that's the pragmatic difficulty, but there is one method in research. It's called trial enrichment. By that, I mean, we might be able to recruit people at an extremely high risk for dementia, characterized by family history, lipoprotein E4 presence, and plasma biomarkers. And by doing so, we may be able to, we're not sure, there is a possibility that we may be able to see a difference within a relatively short time frame. Still, it will take years. But in the first phase, we aim to do a pilot study that can assess the tolerability, safety, and feasibility of fasting in this age group and in this population. But as a first step, what we are doing right now is doing a community survey to design the duration of fasting and the duration of cognitive assessments. So that as community survey is going on now, and we already received some responses, and the participant recruitment is going on. So if you are interested, or if you know someone who might be interested in joining this part of the survey, this is the email address people can contact, and these are the criteria. So there should be a family history of dementia, and they should not have any cognitive syndromes. So please feel free to contact the Academic Unit of Psychiatry of Old Age if you have any questions about this part of the study. Now, food is part of our culture. I mean, in real life, there are challenges. There is a lot of politics and social and economic aspects of intermittent fasting. But I must say, intermittent fasting doesn't mean that we can't attend. It doesn't mean that we can't attend parties or can't celebrate birthdays. I mean, people can practice fasting at varying periods, and so, for example, they can fast during weekdays and eat as normal during weekend. But there are so many aspects in intermittent fasting. So to conclude, some aspects of aging, they show non-linear phenomena with exponential changes in middle age. It's much more than what we thought before. Intermittent stress in general is adaptive. And same applies to exercise, which is another intermittent stress. When we do aerobic exercise, it acts as stress for muscles and blood circulation. So these types of intermittent stress produce adaptive cellular responses. The physiological system undergo a kind of remodeling, and that is adaptive. And that enhances our survival and fitness. a wealth of empirical findings from basic science research, but the limitation is that they are mostly from animal research, and we don't know how it is going to work in humans, and that's why we need to test it out. And we are going to test this hypothesis that it might be beneficial in reducing the risk of dementia. So mechanistically, intermittent fasting has a broad cellular and metabolic impact, which may, favorably affect the dementia pathology in risk reduction. Now, before stopping, I need to show this slide. This is very interesting because this is counterintuitive though. So in Sweden, this Dr. Lars Bygren, he studied food habits of generations, and he meticulously collected agricultural records and traced generations back. And in one northern suburb in Sweden, he studied people's longevity and lifespan, and this happened. He looked at the periods of famine and in 19th century, and what he found was the boys who enjoyed food availability and abundance during the pre-pubertal phase, they had grandsons who had lifespan 6 years shorter than the grandchildren of starved boys. So this food deficiency occurred during their pre-pubertal phase. But the opposite happened when there was food deprivation during pregnancy that adversely affected babies. But when it happened during the pre-pubertal phase, they had grandchildren with longer lifespan. Now, the way researchers try to explain this is transgenomic change or epigenetic changes. So this lifestyle and its effects, nutritional deficiency, sometimes exercise, produce changes in epigenome. As I mentioned, DNA methylation and several others epigenetic modifications. They influence gene expression. Now, the most important thing to keep in mind is that these changes are heritable. They are transmitted across generations, though it is not part of the genome that codes for proteins. The epigenetic changes can be transmitted. So when one does this sort of practice, lifestyle, aerobic exercise, or intermittent fasting, that person influences not only his health, but also health of generations to come. So that's where, another aspect or dimension of this lifestyle lies. With that, I conclude, and I thank Professor Nicola Lautenschlager, who always supported the study and provided guidance, and can't thank her enough. And same with Catherine, who is here. Then Jenny Southam, our research coordinator, our endocrinologist who is involved in the study, Associate Professor Priya Sumitran, and the staff at AUPA, Nicola and Luella, for their tremendous support in carrying the study. I must also thank Serena Navarro Research Foundation and Dementia Australia for supporting the study on intermittent fasting. Thank you for joining me, and happy to take questions.
Fantastic. Thank you so much, Albie. Really interesting presentation. I think I nearly said a lot of food for thought. I'm going to say it out loud. Terrible pun, and it wasn't intended, but I think there's a lot there to think about for midlife and even earlier in life. So I'm just going to open the chat now, Albie, and see if we've got any questions come through. If you do have a question, please feel free to raise your hand as well. Thank you, Amanda, for a lovely comment there for Albie. And Magnolia, we've got a question here, two questions actually, and I'll start with the first. in intermittent fasting, what's the caloric restriction for the periods when people are allowed to eat? Albie, and also a question about your trial, how long will your trial follow-up period be? So maybe if you could give us a little bit of insight there.
Yeah, thank you for these questions, Magnolia. And so, what is a caloric restriction for periods when people are allowed to eat? I'm not aware of any quantification there. What I know is that during the period of eating, that eating window shouldn't have a compensatory eating. That should be the same amount that we eat normally. So once you fast, then you eat the same quantity that you would otherwise eat. The problem with fasting comes when people, it's not an automatic thought that, we are fasting so we can eat more. That brings the problem. But I am not aware of any caloric, in certain figures or in quantification. And how long will the trial follow-up period? So we will do this, the pilot study for 6 months, and then probably the randomization and the proper trial for 2 years. And then we can follow up for any length of time for cognitive decline and also some biomarkers of Alzheimer's disease.
Great, thank you. Any other questions for Albie? Okay, so yeah, here you can see this Albie from Vedran. Maybe if you can read the question for us before you address it.
Yeah, fasting mimic diets like keto, paleo, and carnivore. Could these influence results? Enhance the benefits of fasting? Yeah, keto diet, it's it's artificial, and we know that ketone bodies are very minimally elevated during intermittent fasting. That is natural and physiological, but keto diet is artificial. Now, from few reports, I know that keto diet had some additional impact on cardiac rhythm, and in a very few instances, there were serious cardiac arrhythmias associated with keto diet. But that we don't see that with fasting associated ketone bodies. Whether keto diet can have the same effect, theoretically, yes. And in some studies, investigators used keto diet to measure the same effects that were measured in studies that used intermittent fasting. So there are some similarities, but there are differences as well. But the main benefit of intermittent fasting comes from the stress on cells and the cells' adaptive responses that are stimulated, which doesn't happen with keto diet. And a lot of other things. For example, I said brain-derived growth, neurotropic growth factor changes. That all happen with intermittent fasting, not with keto diet. But keto diet, again, in some neurodegenerative diseases, they found that it can protect neurons, because when glucose is depleted, neurons can utilize ketone bodies.
Fantastic. Thank you, Albie, and thank you for that question. And also thank you to Deb, who's just effectively asked the question that was going to be, I was going to take a little bit of a moment to ask. And are there sex differences between women and men in relation to the study and hormonal influences that you mentioned earlier on women, particularly in midlife and during menopause?
Yeah, so the hormonal changes, in that paper that discussed changes in middle age, their argument is the impact on brain and the brain aging is influenced by so many peripheral changes, the hormonal changes associated with the menopause, and some inflammatory markers, for example, monocyte and T cell infiltration, all impact brain aging. So there are gender differences and changes in estrogen and associated vascular risk. This all can contribute to brain aging and cognitive performance. But fortunately, we can modify these changes favorably so that we can either reduce or prevent cognitive decline in the next few decades. So though there are gender differences, they are amenable to these sort of lifestyles.
Great, great. Thank you. And what about different benefits or effects from the timing of the eating window question here, you know, such as early eating window or later eating window, closer to bedtime, for example?
Yeah, so there is no information about the differential impact of fasting at different times. So people can fast at any time. The only thing that matters is the time duration. So, but if you look at the diurnal variation of hunger, it peaks during evening and it's least in the early morning. So what I've seen is that many people they skip breakfast. That itself is contentious because the studies are divided in findings about skipping breakfast. But we need to thoroughly analyze the methodology and approaches taken by these studies. But many people avoid breakfast, and that helps them to practice fasting for, say, 15, 16 hours. And people find it easier to do fasting when they skip breakfast. But otherwise, there is no differential impact according to the timing of the day.
Very interesting.
Yeah.
Another second question here, which I think is another interesting one. You mentioned metformin. Do you think it can be taken? Obviously, we're not giving any medical advice here, just, you know, your interpretation of the literature. Could it be taken as a preventative medication for anti-aging effect, particularly those who are not able to sort of sustain that intermittent fasting for a long time?
Yeah, first of all, I must say I'm not an expert on the endocrinology or these endocrine diseases. My specialty is different. I just shared, you know, my knowledge about metformin, but that should not be taken as a, you know, a specialist or an authoritative comment. I mean, I shared that as an incidental thing along with effect of intermittent fasting, but you know, we conduct a study with specialist endocrinologist. So I won't comment on that as an expert. Yeah, metformin has, you know, broad effect. And I mean, it's open to anyone who is interested in studying this medication.
Have we got any other questions coming through? If you have one, feel free to whack one in there. Linda has just done that for us. Is there an ideal length of the time to fast?
Yeah,
Thank you, Linda.
no, there is no ideal length, but we know that the glucose store gets depleted. It's not depleted, it's diminished by 12 hours. And then, sorry, the glycogen storage diminished by 12 hours. And by 14 hours, the cells start using ketone bodies. And so 14 hours is the bare minimum, but some others recommend 16 hours or at least 15 hours.
Okay.
That should do the job, but you know, a bit longer may be better. We don't know. In many studies, participants had 24 hours fasting. So in studies, the duration of fasting varied. I can't say there is an ideal length, but 16 or 15, 16, 18 could be sufficient.
Excellent. Interesting. Also, again, you know, being careful, you know, that we don't have a great idea yet around aging and the age that people should be doing this, the impact of food in early development. So we certainly don't want to be, you know, offering up any advice here that is potentially damaging to anyone. But do you have a sense of the age in which starting intermittent fasting might be a good idea? Are we thinking midlife? Are we thinking slightly earlier? What's your instinct and interpretation of the literature so far, Albie?
Yeah, so with this new paper and emerging findings, I think I'm inclined to say that we should start earlier as possible, you know, maybe around 40 years. So earlier is the better, there's no doubt about it, because all these changes happening are, you know, they're cumulative, but they're non-linear. So, for example, some cognitive functions may decline after 40 years. If you look at the connectome changes, they show nonlinear trend with a breaking point around 50 years. So, and the effects of intermittent fasting are, it takes time, for full benefits, so maybe years. So that's a rough estimate.
Okay, thank
Yeah.
you.
And there's one more question, and how often? So there is a popular pattern, 5 days fasting and 2 days eating as normal. So that is 5:2. That's probably the most popular type of intermittent fasting.
Right, thank you. Any other questions? No? Okay. I think that's been a really rich discussion there at the end of the presentation, and certainly a wonderful presentation full of a lot of up-to-date evidence and some thought-provoking evidence as well. So thank you very much, Albie. Very much appreciated.
Thank you. Thank you again, Catherine. Yeah.
I'd just like to draw everyone's attention to our next Narian Academic Unit for Psychiatry of Old Age Seminar in Aging, and that will be by Dr. Sophie Haywood. And Sophie will be presenting on trauma-informed principles of safety, trustworthiness, choice, collaboration and empowerment in physiotherapy and in multidisciplinary rehabilitation. That will be on the 14th of May. Sounds extremely interesting, I think. So thanks again to the organisers behind the scenes, Jess and others who do so much. And thank you all for attending. I'll close the meeting now. Thank you.
Thank you.